Hello and welcome everyone. This is the first podcast of day 31, chapter 27. In this podcast, we're going to cover Objective 1, structural and functional adaptations that have contributed to the success of prokaryotes. So let's go ahead and get started. All right, as we begin to look at the information in this chapter, I think it's important to put this group called prokaryotes in its proper place. We know that the Earth is roughly 4.6 billion years old. Our best estimates are that bacteria arose, or I should say prokaryotes for right now, arose 3.5 billion years ago. A couple things I want to point out as we look at this figure here is that even though they arose around 3.5 billion years ago, we suspect they formed before then. This just means we have scientific evidence that they were around 3.5 billion years ago. More importantly, as we look at this diagram, we see that the prokaryotic group here continues to evolve. They are not the same as they were 3.5 billion years ago. Because of their small size and their rapid reproduction, they can rapidly obtain new adaptations to allow them to live in the most diverse environments of any organism or group of organisms on this planet. They can live in very salty conditions. They can live in dark conditions, high pHs, low pHs, environments where there's no oxygen, environments where there is oxygen. They can live in the dirt. They can live within other organisms, live within ourselves. They're an incredibly diverse group of organisms. You could bend down and pick up a handful of soil, and there's going to be more prokaryotic organisms in that handful of soil than all the humans combined that have ever lived on the planet from the beginning. Well, it's from the beginning of humans, which we'll come back to in a moment. So they're incredibly diverse and they're incredibly numerous on this earth. And they play a very important part of our ecosystem. And we'll talk about that in our very last objective of this chapter. So they continue to evolve. Just like animals, when they have formed less than 1 billion years ago, they continue to evolve. And we should look at this lonely group up here, the humans. It's important to note, and this figure does a very nice job of showing this, is that we humans have really occupied this earth for a very, incredibly small portion of time that Earth has been around, and an incredibly small portion of time since the first evidence of life here at 3.5 billion years ago. So we're going to talk about all of life that has formed, and we're going to try to give them equal time, but recognize that the prokaryotes, even though they get only 1 day, they really have been around much longer than any others and are much more diverse than any other group of organisms on Earth. All right, now I'd like to try to get us out of the habit of using the term prokaryotes. I used it on the last slide because that's what the figure showed, and I'm going to slip and every now and then use it because it's been part of my vocabulary for a very long time. But we've also known for quite a long time that the term prokaryote is a little limiting because it lumps these two groups of organisms, bacteria and archaea, into the same group. However, we know that bacteria and archaea are incredibly different. We know that many features of archaea are more similar to eukaryotes, you and I, and all of animals and plants and fungi and protists. Some features of archaea are more similar to bacteria. So they really deserve to be their own domain. And so we recognize 3 domains. Bacteria, archaea, and eukarya are the domains of life. If you ever use the term prokaryotes, people will know, scientists will know that you're talking about bacteria and archaea, but we really want to get out of the habit of using that term prokaryotes. So as I mentioned on the last slide, bacteria have been around for 3.5 billion years, and throughout this long time they continue to evolve. And they continue to gain new adaptations to survive in the very diverse environments that they inhabit. Despite being around for 3.5 billion years, we can lump bacteria and archaea shape into one of three categories. They can be very spherical, very round, so we can call them spheres. A better scientific way to describe this would be cocci. They can be rod-shaped, like bacteria, like E. coli. And a better way to describe this scientifically would be bacilli. And the final shape would be spiral. This shape of archaea and bacteria that we just spoke about is maintained by bacteria and archaea having a cell wall. This cell wall can do several things. It helps maintain its shape of the cell. It protects the cell and prevents it from bursting. Now, other organisms also have a cell wall. So plants and fungi, they also have a cell wall, and their cell wall is made up of cellulose in plants or chitin in fungi. And we'll talk about that in later chapters. However, bacterial cell wall is made of something else called peptidoglycan. Archaea, again, this is one of the differences between bacteria and archaea, contain various polysaccharides. Remember, cellulose is a polysaccharide, but in archaea it's not cellulose, it's another polysaccharide. And various proteins. All three of these function as a cell wall and functions in these categories of maintaining a shape, protecting, and preventing the cells from bursting. But they're made of different components. The next thing I want to talk about is the Gram stain. Your book goes into some detail on how a Gram stain is done. I don't want you to worry about that. You'll learn that when you take microbiology. What's really important is how we use the Gram stain to classify different kinds of bacteria. Bacteria, not archaea, bacteria can be classified as either Gram positive or Gram negative. Gram positive bacteria have cell walls that are simpler, so simple cell walls. And these cell walls contain more peptidoglycans. Gram-negative bacterium, the cell walls are structurally more complex. And they have less peptidoglycans. Gram staining is, as I said before, is a useful way to classify the different kinds of bacterium based upon their composition of their cell walls. However, Gram staining also has a valuable clinical purpose, a medical purpose. It's used in medicine because we can quickly identify whether or not a bacterial infection is gram-positive or gram-negative. Depending on if it's gram-positive or gram-negative, different kinds of antibiotics can be prescribed. The structural complexity of a gram-negative bacterium allows it to respond differently when it's infecting us. They tend to cause higher fevers, can lead to shock, and are generally more toxic. This complexity also allows gram-negative bacteria to evade our immune system. I'm running out of room here on the side, on the bottom, so I'll write it over here, but they also tend to be more antibiotic, AB for antibiotic resistant. A gram-negative bacteria infection is much more difficult to treat than a gram-positive bacterial infection. And again, this is due to the fact that they are structurally more complex, their cell wall is, and thus it's harder for the antibody to penetrate the cell wall of the gram-negatives. Now, it is true that gram-positives are typically easier to treat with antibiotics. It should be noted, however, that many gram-positive bacterium have become resistant to typical antibiotic resistance, and they require higher levels of antibiotic treatments and multiple antibiotic treatments. So some of the more potent Staphylococcus infections and MRSA that you may have heard about in the news are gram-positive bacterium, but they cause a lot of problems because of their resistance to antibiotics. But still, you should remember that gram-negatives tend to be tougher bacterial groups to fight with typical antibiotics. Okay, the next thing I want to do is talk about these terms capsule, fimbriae, and pillae. And I'm also going to throw in endospores as well into this discussion. These are all features of bacteria that have, I should say they are adaptations of bacteria that have helped them survive. So let's first begin with a capsule. So many bacteria, we could draw it maybe like so. This is the cell wall. Many bacteria surrounding this cell wall will have what's best described as a sticky layer of sugars or of polysaccharides. So we have this sticky layer that surrounds it. So we'll just draw it in blue, the squiggly line. And these are sugars and proteins. Large sugars, so not simple sugars, but they're like polysaccharides. And this is our capsule. As your book says it, a sticky layer of polysaccharides. And proteins. Capsules have a few different purposes, and so let's go ahead and list some of them here. It allows them to adhere or stick to substances, or other bacteria. They can also protect against dehydration, and protect against a host immune system. So this is the first adaptation that have helped bacteria survive for 3.5 billion years. Another way that bacteria can stick to substances are by using these other structures called fimbriae. Figure 27.6 in your book shows a really cool picture of what the fimbriae look. But essentially what it is, if we were to draw a rod-shaped bacterium here, whereas the capsule is this kind of sticky stuff all around the cell, fimbriae almost appears as these very sharp spikes that come out of the bacteria. You may be familiar with the terms cilia and flagella, and we'll talk about those in a moment. That's not what these are. These are fimbriae, and they help the bacterium stick to other bacterium and to surfaces. One really interesting example of it would be the bacterium that causes gonorrhea. These fimbriae allow it to attach to the cell surfaces of the host organism. The other structure is called pillae. These resemble fimbriae, except they are less numerous. So here are two bacterium, and pillae between these two will allow these two bacteria to connect with each other before they allow DNA transfer. And we haven't talked too much, or maybe even at all, about this, but we have DNA in these bacterium, and the pillae allow them to attach, and when they attach to each other, we can have DNA transfer. So when they come together, the bacteria come together, the DNA can transfer from one to the other. They are often referred to as sex pillae. And figure 2712 in your book shows a good example of what this looks like. Now, another way bacteria protect themselves is by forming structures called endospores. When the bacterium are presented with a lack of nutrients, they form endospores. And when they form this endospore, what they do is they copy their DNA and then surround the DNA with a tough multi-layer structure. Multi-layer coat, let's call it. Once this is formed, water is removed, metabolism shuts down. Then the endospore is released. These endospores can remain for centuries, so for hundreds of years they can remain. You can place them in boiling water and that often will not even kill them. It requires the intense heat that we find in an autoclave to typically destroy an endospore. So just two more things we want to talk about: taxis and binary fission. And these won't take too long. So taxis. This is just the ability to move. Approximately 50% of bacteria do this. That is, can move using their own structures. This is very advantageous for them because it allows them to move towards nutrients, or they can move away from danger. For instance, like a toxic substance. The most common way that bacteria undergo taxis is using a flagellum. We see flagellum in eukaryotes as well, but structurally and functionally, the flagellum of bacteria and Archaea are similar to each other and different from those that we might find in eukaryotic cells. Alright, the last thing we want to talk about is binary fission. Alright, so what is binary fission? Binary fission is how a bacterial or an Archaea cell takes its genetic material and copies it and produces it into two new cells. Essentially, it is mitosis. So you have your original cell here with its DNA in it. It copies the DNA, so you have two copies of it, essentially two identical copies. And then the cell divides, each with its copy of DNA. So if you have a single bacterium cell, when it divides and forms a colony, we would consider that colony to be genetically clones of the original one. And that's all you need to know about binary fission. That ends the podcast for Objective 1. If you have any questions, please let us know. Thank you. Bye.